Hi, and welcome to video 2.3. In this video, I'll be talking about the naming of nucleosides and nucleotides, breaking down the differences in the types of bonds, especially the phosphodiester bonds and phosphoester bonds that are quite, challenging to say, but not too bad to understand. With that in mind, let's dive deeper into the naming of nucleic acids. Here, as an overview from the last video, I've made a small table. We have the base, so the nucleic acid, and I've put guanine and cytosine for purine and pyrimidine. We'll cover the rest as well. I've also named the nucleoside. Remember, the nucleoside is when you have not just the nucleic base, right, or the acid, so to speak, but we'll just call it base to make it simpler. Nucleic base. And what we do is we will just, add the sugar to it, the ribose sugar. So with the ribose and the base, you get the nucleoside, and there's a different name. With guanine, remember the purines are the two rings, the purine pyrimidines are just one. As a reminder, guanine has a double bond to oxygen, where adenine does not, right? And that would be the other purine there. Adenine and guanine. Guanine turns into guanosine. So this osine name, it tells you it's a nucleoside, right? If you have the sugar, if you just have guanine by itself, it's guanine. If it's attached to the sugar, it becomes guanosine. And that same naming convention works for adenosine. Adenosine, right? Adenine turns into adenosine. For cytosine, again, the base, when we add the ribose sugar to it, right, we add adene. So that's for the pyrimidines. So cytosine, thymine, and uracil are our three pyrimidines, which turns into thymidine and uridine. Thymidine and uridine. Right, so good names to keep in mind, right? These patterns. If you call guanine by itself, with the free chemical guanosine, that's incorrect. With the nucleotide, right, this is where you're going to actually add the phosphate as well. And then the name includes the phosphate. So the simplest nucleotide, for example, if I was to take, and I'll do one example here with, let's say, guanosine. I take a sugar. There's my ribose sugar, and it's connected to guanosine, right? Let's call that carbon 1, 2, 3. I put an OH here, right? It's DNA, so there's no 2' OH. That's how you know it's DNA. And this is now connected, right? Carbon 4 and 5 to a phosphate group, right? Without the phosphate, this whole thing is called guanosine. This is called guanine. And this is now the full complete nucleotide. We call it guanosine monophosphate. Now keep in mind, there's an important distinction, as this is slightly inaccurate, right? Guanine is a DNA base that can be present in DNA or RNA. Remember that it's only thymine and uracil that are only found in specific ones, right? Where thymine is only found in DNA and uracil is only found in RNA. In the case of guanine, it's found in both, and so this isn't actually accurate enough. We need to be very specific and say, hey, there's no 2' OH here. So this is actually deoxyguanosine, right? For thymine and uracil, as you'll see in a practice question to come, you won't need to worry about this, right? But for guanosine, deoxy tells you there's no OH there, deoxy for DNA. So this is the name of the nucleotide. For a pyrimidine, we can do something very similar, right? We take a sugar. Let's go ahead and do uracil, right? Uracil is only found in RNA, and so I'll have the OH there. There's my U for uracil. And let's now connect it to two different phosphates. So what we have now is uridine diphosphate, right? Monoditri for the phosphates. Right? We don't need to put deoxy, right? There is an oxygen there, so we shouldn't put deoxy. And if there is an oxygen, you just leave it, right? So uridine diphosphate. This is how we name nucleotides. And the important thing, again, if you have DNA, you have to add deoxy to the end of it. So here are a couple practice problems. Feel free to pause the video and go back if you need to look at that table and try to name these two chemicals. Now that we've done that, if you're done, and good luck with the work, let's go through it. So for the first one, this is going to be, again, we know it's a pyrimidine because there's one ring. We see a methyl group, right? That tells us that it's thymine. Thymine is unique for the methyl group. And we know it's connected to the sugar, so we're not going to call it thymine. We're going to call it thymidine. Right? There's no phosphate, so this is simply thymidine. Right? If you want to be more accurate, you can call it deoxythymidine because there's no OH here. However, thymine, if you remember, is only ever present on DNA. So it's actually arbitrary. In the case of thymine, you don't need to put deoxy here. It's advised, maybe you should, just to keep it consistent, since there is no oxygen here. But in biology, we don't see RNA form of thymidine, at least that I'm aware of. For the second problem, we see now, once again, it's another a pyrimidine, so it's one ring. This one is going to be uridine. Actually, is that uridine? Let's go and check. Right? I think I actually made a small error here. My apologies. On the structure here, there's supposed to be a double bonded O there. So I'm gonna run with this take still, but this is actually supposed to be thymidine with a double bonded O there. Keep that in mind as you do this. This is the same thing, just with uracil. That's why they looked a little different. And if you caught that before I got to it in the video, good work. Right? Thymine is going to need that second OH there, or that second double bond oxygen. Here with uridine, right, we're going to also call it uridine because it's the nucleoside, but we also have the phosphate, so it's a nucleotide. We need to add the number of phosphates. To do that, I'll just pull this here. Uridine monophosphate is the name of this chemical. So again, the hard part of naming these is recognizing, right, what DNA base you're dealing with. And when you pause the initial video, right, since that double bond was missing, don't be upset if you couldn't find that one. The structure is very key, right? So make sure that you're, able to identify from a list, if you look at them visually, what those purines and pyrimidines are. And then make sure you're using the name correctly if the sugar's attached, right? Again, that name being here. Feel free to take a screenshot of this if you want for your own naming. Now, looking at the function and structure of these nucleotides, the complete building block of nucleic acids, it depends on the number of phosphates. So monophosphates that have a single phosphate group, right? These are typically used, if we think about their function, right? They can just be free floating, so they can be around, right? So for example, we have ATP, which is adenosine triphosphate, which we'll talk about soon, but there's also something called AMP, adenosine monophosphate, right? Monophosphates are just around. They can be, free molecules, and we'll talk about their, I'd say they're a sign of low energy in the cell, right? Because ATP will form these things when ATP is high energy. But we'll come back to this much later in section 7. I don't want to dive into here too much. The much more important function of monophosphates is the fact that they are the building block for DNA and RNA polymers. Right? That long strand is built with one phosphate and then another sugar and then one phosphate. So these monophosphate nucleotides are critical as they are the building block of DNA and RNA polymers. They also exist in other places. I just wanted to quickly touch on that. But really understand that these monophosphates, right, without the phosphate, these things cannot be incorporated into DNA polymers, right? We need them to store information. So very crucial function. Talking about triphosphates now, just jumping to them, right? And I guess, actually before I jump to triphosphates, I do want to talk about one specific thing here, which is that these monophosphates contain a specific bond. So apologies for jumping around. The bond we're looking at that I want to talk to you here about is this one right here. We call this a phosphoester bond. Right? This is not a diphosphoester bond. It's just a singular phosphoester. The idea here is that in organic chemistry, an ester is when a carbon is connected to a double bond to oxygen, an oxygen connected to some sort of other carbon, right? That is an ester group. Here we can see that if I just replaced this carbon with the phosphorus, I have this pattern, right? Phosphor, double bond to oxygen, single bond to oxygen, connected to some sort of R group, right? The sugar. So this is basically an ester bond right here, but we call it a phosphoester because instead of carbon, it's phosphorus. Useful to call this now, and why I wanted to come back up here to bring it up, is because we're going to see multiple of these together and they get different names. So this is the first kind of new functional group to keep in mind. Phosphoesters are present in monophosphates. When we go to the triphosphates, they're going to contain, similarly, these phosphoester bonds. Right, so here I see a phosphoester bond. Right, same place. We see the phosphor, it's connected to the oxygen and the OR. The other thing, though, that they're going to have is phosphoanhydride bonds. Right, now in organic chemistry, an anhydride is when you have two carbonyls linked by an oxygen. So considering this, look what we have here, right? With these three different phosphate groups added on, right? Phosphate 1, phosphate 2, and phosphate 3. So I'll kind of number them as distance from the sugar. We can see that they're connected to each other. And I want to put a highlight on this blue box, right? If these phosphors were converted to carbon, we'd have this kind of anhydride bond. Right? That's a functional group from OChem. So here we have an anhydride bond. I like to call it a POP bond because it's POP. Right? These are phosphoanhydride bonds. Right? There's one here, and there's also another one over here. So keep in mind that these aren't all linked by phosphoesters. In this case, we have one phosphoester and two different phosphoanhydride bonds. Why does that matter? If you look at this phosphoanhydride bond, there's lots of negative charge, right? So there's two negative charges here, one here, one here. Lots of negative charge. It's very unstable. And the thing about anhydrides is they're easily broken with water. And so this is also true here. Water can break this bond, and what it does is it releases a lot of energy. So these, and for example, ATP, adenosine triphosphate, these are stores of energy ready to be used. Right? If water comes in or something else comes in and breaks that bond, those negative charges get to be free of each other. It makes a lot of extra stability, and that energy has to go somewhere, so it produces a lot of heat. Right? These things break off, and that energy or heat is used to power all these different processes in the cell. So again, key difference in the nucleotides. A monophosphate is structural. It builds DNA and RNA. A triphosphate with those phosphoanhydride bonds is a way to store energy and break those bonds later to use the energy to do things, and that's metabolism. Putting it all together, feel free to take a look at this last question, and you can pause the video to get the name and all these different factors about this nucleotide here. Hope you had luck with this question. Let's go through it. The name of this would be, and I'll do it in red, deoxyadenosine triphosphate. Right, we know it's adenine here because there's only a nitrogen. There's no double bond like guanine, and it's 2 rings, 3 phosphates, and there's no oxygen on the sugar. That's why it's deoxy. In terms of the number of H-bond donors, we have 2 here. We have 1 here. So there's 3 H-bond donors. In terms of the number of H-bond acceptors, we have quite a lot. So going through it, remember that these ones both count, since the nitrogens have double bonds, where this one doesn't count, because it's near a ring and can do resonance. This one will count. This one will also not count, because it's, again, when you see that N has a double bond, it counts. When N only has single bonds, it doesn't. So that's 1, 2, 3. And then oxygens are going to be much more simple. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31. So pretty ridiculous amount of H-bond accepting going on in this molecule, which can tell you that, it's going to be pretty water soluble. It's going to want to really find those waters. In terms of the number of phosphoester bonds, right, we only have the one, right? There's only ever one phosphoester bond that's connected to the sugar. And for the phosphoanhydride bonds, we're going to have two of them connected to the phosphates. So, hope you got that question correct. And that really finishes up the naming and structural part. Now we're going to focus, moving forward, on the polymerization of nucleotides, making DNA and RNA polymers, and understanding how they work. I'll see you in the next video to discuss.